One of the posters that I'm presenting here at EHA is the DREAMM-6 preliminary trial results, which is looking at a combination of Belantamab Mafodotin in combination with standard of care regimens. So Belantamab Mafodotin, which is also known as Belamaf, is an antibody-drug conjugate that targets BCMA. In the pivotal DREAMM-2 clinical trial, the response rates for a very heavily pretreated and refractory population was a response rate in the region of 20 to 25%. And the median duration of response for the 2.5 milligrams per kilogram cohort was 11 months. So the logical next step is to combine this with standard of care agents, and that's what we've done in DREAMM-6. There's two parts to this. There's the part A, which is a combination with Revlimid and Dex, and part B, which is a combination of Bortezomib and Dex. And in this poster, we're focusing on part B. So this is for patients who had relapsed myeloma. They needed to have at least one prior line of therapy. And I should point out that we didn't exclude patients who were either refractory to Bortezomib or to Revlimid. So we're reporting predominantly the 2.5 milligram per kilogram cohort of Belamaf in combination with Bortezomib and Dexamethasone because this is the most mature data. The treatment schedule was for patients to receive the antibody once every 3 weeks, and then in combination with Bortezomib, which is as per license, which is 1.3 milligrams per kilogram, days 1, 4, 8, and 11, again every 3 weeks. They receive 8 cycles of the triplet combination, and then you go on to receive Belamaf monotherapy until disease progression or intolerance. So we're reporting on 18 patients in this particular cohort. They had a median of 3 prior lines of therapy, and about 40% had more than 4 prior lines of therapy. What we can see in this cohort in terms of adverse events was that all of them experienced some sort of adverse event, which is what is really expected. But just in terms of focusing on the adverse events of special interest, there was quite a low incidence of infusion-related reactions, which is in keeping with the previous studies. And just to remind everyone that we don't use mandatory pre-medications with Belamaf infusions. The second event of special interest is thrombocytopenia, because this is really an overlapping toxicity between the two agents. And we did see quite a high incidence of this. It was predominantly grade 3 and 4. However, we were able to manage this predominantly with dose interruptions, dose modifications, and the occasional platelet transfusion where required. And then finally, we know that corneal events or keratopathy is a feature of Belamaf, and all patients did experience some form of keratopathy. It was predominantly grade 2 and grade 3, and again, as per previously, it was managed by interruption of dosing, dose modification, and the judicious use of preservative-free eye drops. Now that's really key to managing the eye toxicity. We didn't see any grade 4 events, and no patients discontinued Belantamab due to any sort of adverse event. What was encouraging was when we look at the efficacy, was that the overall response rate was 78%. And more interestingly, the VGPR rate and above was 50%. So this group of patients were achieving quite deep responses. So in summary, this is preliminary data, and we will be following this up in the future. But certainly the toxicity profiles appear to be quite manageable, and the responses are quite encouraging for a heavily pretreated population. If you want to benchmark this against historical data for Bortezomib and Dex in the same setting, we'd be expecting a response rate in the region of 50 to 63%. So this is certainly quite encouraging for the triplet combination. And we hope to be reporting on other aspects of the trial later on in this year. The other two posters I'm reporting at EHA with Belamaf are looking at the quality of life and also an indirect comparison to work out a comparator for Belamaf in the relapse and refractory setting. In terms of quality of life, we looked at the DREAMM-2 clinical trial, which was the phase 2 study of Belamaf for patients who were refractory to a proteasome inhibitor, an immunomodulatory agent, and were either intolerant to or refractory to a CD38 monoclonal antibody. Now, those results have already been published and are being updated at this year's Congress by Dr. Loniel and other colleagues. But in terms of quality of life, we assessed patients using the standard cancer tools for quality of life assessment, as well as myeloma-specific EORTC quality of life tools. What was quite interesting was that overall there was an improvement in health-related symptoms. And this was specifically seen in functioning, fatigue, and bone pain. And I would think that in a very heavily pretreated population, to see an improvement in fatigue is actually quite difficult. And certainly some of the trials that I've been involved in previously have not been able to show that. Also, typically what we see in clinical trials is that there's a deterioration in quality of life as time goes on, as patients tend to progress. Now, we didn't observe this, and overall the quality of life was improved in the Belantamab arm. So I think that overall that this is a manageable treatment for patients, and their quality of life does seem to improve, particularly for those patients who do respond to treatment. Now, if I mention the second poster, this is looking at an indirect comparison. Now, the reason that it's important to perform this is that the phase 2 clinical trial, which is DREAMM-2, was a single treatment arm, in that it was Belantamab in both arms. We did look at 2 doses, the 2.5 milligram per kilogram dose and the 3.4 milligram per kilogram dose, but there was no comparator. Now, this becomes important as people want to know what to benchmark the results against, and particularly when it comes to licensing and reimbursement, there needs to be some sort of comparator. So in this abstract, we performed a analysis of other clinical trials which may report similar sorts of populations. And through a literature review, the only study which really came close was the STORM-2 clinical trial of Selinexor and Dexamethasone for patients with relapsed and refractory myeloma. So within the 2 cohorts, we identified a population of patients and matched them in terms of as many demographics as was possible. Now, of course, we were unable to do that across the board. And that is one of the limitations of studies. So, for example, for cytogenetic analysis, extramedullary disease, we were unable to match them because that data was not available. But nevertheless, we then ran the response rates, progression-free survival, duration of response, and overall survival analysis for both. What we saw was that the overall response rates were very similar for Selinexor versus Belantamab for a similar sort of population. What was different, however, was the median duration of response and the median overall survival, which was superior for Belantamab versus Selinexor. So whilst we can't draw firm conclusions from this, this indirect analysis does show some potential advantages of Belantamab over Selinexor within this heavily pretreated population of patients.